United Kingdom and has expertise in the fields of gene technology, protein technology, and bioinformatics. She has research experience working in various projects in the areas of phylogenetics, industrial biotechnology, clinical research, molecular modeling, etc., and has also presented seminars in varied domains. She is also having industrial exposure working in reputed companies in the fields of bioinformatics, bioengineering, and next-generation sequencing technologies. I welcome Ms. Maithili Verma to deliver her talk on Illumina sequencing and its applications. Over to you, ma'am.
Thank you so much for that welcome. And good evening, everyone. I would like to thank Mr. Thiru for giving me this opportunity to speak on this esteemed platform. And I will share my screen now. Shall I start?
Yes, you can start, ma'am.
Okay. I will be presenting Illumina sequencing and its applications. And this will be the outline of my talk. I'll be talking about an introduction to DNA sequencing and the history, Illumina sequencing principle, the DNA library preparation and the kits used, the applications of DNA sequencing, and the types of Illumina sequencers and their applications. So coming to the definition or the introduction to DNA sequencing. What is DNA sequencing? It's a laboratory technique that's employed to know the correct DNA sequence by the sequential chemical reaction. That is known as DNA sequencing. And there are two key processes in DNA sequencing. One is laboratory processing and computational analysis. So once the chemical reaction is completed, the machine-generated data is sent to the computer lab. So what are the steps in DNA sequencing? This would vary from, this is a generalized representation of DNA sequencing, and it can vary from one platform to another. So the steps include sample preparation, that's DNA extraction, then PCR amplification of the target sequence, amplicons purification, sequencing pre-prep, then DNA sequencing, and finally data analysis. So coming to a brief history of sequencing, there are three generations of sequencing. The first generation of sequencing was called Sanger sequencing, and it was developed by Frederick Sanger in the year 1977. This technique involves the incorporation of chain-terminating dideoxynucleotides by the DNA polymerase during in vitro DNA replication. So here you can see this is a normal nucleotide and this is a dideoxynucleotide. That's one which, or it's also called a chain terminator. So this is, it's a chain terminator because it does not have the OH group here. The OH group would connect the bases together to form a chain. So that is only possible with the normal nucleotides, which have the OH group at this position. So though Sanger sequencing has been replaced by next-gen sequencing methods, especially for large-scale genome analysis, it remains in wide use for smaller scale projects and also for the validation of next-gen results. So coming to the second generation of sequencing, it's also known as amplified molecule sequencing or massively parallel sequencing. And in this, there are examples like the Illumina sequencing and the Roche 454 sequencing. So in NGS, a vast number of short reads are sequenced in a single stroke. And to do this, firstly, the input sample must be cleaved into shorter segments. And the length of each segment will depend on a particular sequencing machinery that's used. So the longer fragments are ligated onto generic adapters and they're annealed to the slide using the adapters. And PCR is then carried out to amplify each read. Then they are separated into single strands and then sequenced. So coming to the third generation of sequencing, this is also called single molecule sequencing or long read sequencing. So it is a class of DNA sequencing that works by reading the nucleotide sequences at the single DNA molecule level. In contrast to the existing methods that require the long strand to be broken down into smaller segments and then inferring the nucleotide sequence by amplification and synthesis. So examples of these are Pac Biosciences and Oxford Nanopore sequencing. So coming to the differences between Sanger and NGS. In principle, the concepts between Sanger and NGS technologies are similar, but the critical difference between the two is the sequencing volume. The Sanger sequencing method only sequences a single DNA fragment at a time, whereas NGS is massively parallel and it sequences millions of fragments simultaneously per run. So the steps in Sanger sequencing. What are the steps in Sanger sequencing? One is DNA fragmentation, then in vivo cloning and amplification of the gene, then cycle sequencing by adding these the polymerase, the DNTPs, and the labeled ddNTPs. Finally, you'd have capillary electrophoresis, and the results are then read and interpreted by a chromatogram. Coming to NGS, here the DNA is fragmented, and then in vitro adapters are ligated to the ends of this fragment. Then there's the generation of Polanyi array, or the generation of clusters of DNA fragments. The last step is cyclic array sequencing. I will be explaining this in more detail in the next slides. So this is the Illumina sequencing principle, and this is a good representation of this principle. First, the flow cell is used, and the DNA attaches to the flow cell via complementary sequences. Then the strand bends over and binds to the second oligo on this flow cell. Then DNA polymerase synthesizes the reverse strand. And you can see here in this image, the complementary strand is being synthesized. Then the two strands release and straighten. Each forms a new bridge, and that's called bridge amplification. The result is a cluster of forward-reverse, DNA forward-reverse and strands, forward and reverse strands. So coming to the basic terms used in NGS. There are certain terms that are very important to understand NGS and or sequencing in general. These are read length. It refers to the number of base pairs that are sequenced from a DNA fragment. And coverage is the number of reads that align to a known reference. Coverage level is also known as depth, the average number of bases that align to the reference bases. For example, a whole genome that's sequenced at 30x coverage would mean that on average, each base in the genome was sequenced 30 times. And at higher levels of coverage, the base calls can be made with higher degrees of confidence. So coming to another term, that's paired end sequencing. This is sequencing from both ends of the DNA fragment in the same run. So coming to the Illumina adapters for the library. So these, the libraries must contain the P5 region and the P7 regions. These regions are necessary to bind to the flow cell and generate clusters on the flow cell. And the libraries must also contain the read1 sequencing primer binding site and the read2 sequencing primer binding site. These are necessary to initiate the process of sequencing. And also the indices 1 and 2. These are required to identify the sample. There are sample identifiers or barcoding sequences. So now I will be talking about each step of the Illumina sequencing principle in detail. First, the first step is to prepare the genomic DNA sample. First, the DNA of interest is fragmented into smaller strands that need to be sequenced. So that can be done through the process of sonication, nebulization, or enzyme digestion. So sonication is through sound, nebulization is a fine mist, and then adapters are ligated to the ends of these fragments. Then you would have to denature the double-stranded DNA into single-stranded DNA by heating it to 95 degrees Celsius. So the next step is to attach DNA to the surface. So here you can see a flow cell. It contains the flow cells contain a dense lawn of primers on its surface. And the single-stranded DNA is then bound to the inside surface of the flow cell channels. Now coming to the next step, which is bridge amplification. Here the unlabeled nucleotides and the polymerase enzyme are added to the flow cell to initiate this bridge amplification process. As you can see here, the strand bends over, binds to the second oligo on the flow cell. The next step is the fragments become double-stranded. So in this step, you would add the primers, nucleotides, the polymerase enzyme, and the buffers to the flow cell. And this would result in formation of the double-stranded DNA. Now coming to the next step, which is denaturation of the double-stranded molecules. The original strand is washed away, leaving behind only the strands that were synthesized to the oligos and attached to the flow cell. So the next step is you would repeat the steps 5 to 7. So the cycle of new strand synthesis and denaturation, it takes place to result in, and it will result in multiple copies of that sequence. That's called amplification. So finally, several million clusters of the double-stranded DNA are generated on the flow cell. Now we come to the sequencing process. The first step in that is to determine the first base. So the first chemistry cycle is carried out to initiate the first sequencing cycle. So you would have to add all the 4 reversible terminators, the primers, and the DNA polymerase enzyme to the flow cell. The P5 region is cleaved off, and as I said before, you would add the primers, the polymerase, fluorescently labeled nucleotides, and the buffer to the flow cell, and the first base gets incorporated. Then you would need to image the first base. So after laser excitation, you'd capture the image of fluorescence from each cluster on the flow cell, and you need to record the identity of the first base from each cluster. So the steps will involve remove the unincorporated bases, you detect the signal, deblock, and remove the fluorescent signal, and initiate the next cycle. So the next step is to determine the second base. As before, you would add all the 4 reversible terminators and the enzyme to the flow cell, and the primers and the buffer are added to the flow cell. The second base then gets incorporated. Now coming to imaging the second cycle, you'll have to collect the data as before and record the identity of each base from each cluster. And the steps are the same as before. That's remove the unincorporated bases, detect the signal, deblock and remove the fluorescent signal, then initiate the new cycle. So in this way, you would sequence multiple reads over multiple chemistry cycles. So you repeat the cycle of sequencing to determine the sequence of the bases in each fragment, a single base at a time. So the identity of each base in the cluster is read off these sequential images. So as you can see here, thymine would be represented as green, guanine as blue, cytosine as red, and adenine as yellow. You would be able to see and understand which bases are there. So finally, you would have to align the data with the reference data. So you would align this sequence with the reference sequence to find out the differences between them. Now I would like to show you a short video of this Illumina sequencing principle so that the concept is clear.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with alon, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized, and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities.
So in summary, I would like to say the Illumina sequencing principle has 4 steps. That's sample preparation, cluster generation, DNA sequencing, and finally, data analysis. So coming to the library preparation, it's a very important step in sequencing. So first, the nucleic acid has to be cleaved to a proper insert size, and then adapters have to be ligated to the ends of these fragments for the purpose of flow cell binding and also for sequencing primer binding. As I said before, these libraries must contain the P5 and P7 regions for the purposes of clustering, and the read 1 and read 2 sequencing primer binding sites for the purpose of sequencing, and the indices 1 and 2. These are sample identifiers or barcoding sequences. So coming into Illumina's library preparation kits, there are 2 ways in which the library preparation can take place. One is through mechanical fragmentation, and the other is through enzymatic fragmentation. So under mechanical fragmentation, there are 2 kits. That's the TrueSec Nano DNA kit and the TrueSec DNA PCR-free kit. The enzymatic fragmentation has 3 kits. That's Nextera DNA, Nextera XT, and Nextera DNA Flex kits. So coming to the mechanical fragmentation, the workflow is as follows. Here the genomic DNA is fragmented. Then the fragments are narrowly size selected, and the adapters are ligated to the ends of these fragments. Then PCR is required to complete these libraries, but only for the TrueSec Nano kit. That's this one. Then coming to enzymatic fragmentation, the Nextera XT kit. This method utilizes specialized enzymes known as transposases. And these transposases simultaneously cut, that's fragment the DNA, and also tag or attach a small section of DNA. And that's this whole process is known as tagmentation. That's a combination of tagging plus fragmentation. And this is followed by PCR amplification. The resulting fragment would have these regions, that's the P5 and P7 region, the indices 1 and 2, and the read 1 and read 2 sequencing primer binding sites. This eventual fragment is ready to be sequenced. Now coming to the Nextera DNA Flex library preparation, this uses a bead-linked transposome. So the genomic DNA then binds to this bead-linked transposome and results in the formation of a DNA BLT complex. The enzyme called transposase tagments the DNA as before, and the resulting, as I explained before, what tagmentation is, and this is followed by PCR amplification. This fragment then has the necessary segment of DNA to be sequenced. So what are the types of library preparation kits? Which version of Nextera should I use? So it depends upon the types of applications that you want to perform. For large and complex genomes, you would have to use, such as humans, mammalians, plants, and invertebrates, you would use the Nextera DNA kit. And for small genomes, such as bacteria and viruses, you'd use the Nextera XT kit. For both small and large genomes, you'd use the Nextera DNA Flex kit. The DNA input would range from 15 nanograms with Nextera DNA to 1 nanogram with Nextera XT and 1 to 500 nanograms with the Nextera DNA Flex kit. So coming to the sequencing applications, NGS technologies have a range of applications, such as whole genome sequencing and targeted sequencing. So under whole genome sequencing, there's resequencing and de novo assembly sequencing. Resequencing refers to the sequencing of a part of an individual's genome in order to detect the differences between that individual and the standard genome of that species. And de novo assembly sequencing refers to the sequencing of a novel genome where there is no reference sequence for alignment. So under targeted exome sequencing, whole exome sequencing, this is a powerful technique that is used for a broad spectrum of heterogeneous neurological disorders. And also under targeted sequencing, there's amplicon sequencing. This is a very targeted approach that enables researchers to analyze genetic variations within specific genomic regions. So under amplicon sequencing, we have mitochondrial sequencing and metagenomics. So mitochondrial sequencing involves the detection and analysis of mitochondrial associated disease variants. And metagenomics, this is another common application in sequencing for the sequencing of bacterial 16S rRNA gene across multiple bacterial species. And it's a widely used method for phylogeny and taxonomy studies. So coming to regulation, the interaction between the proteins and DNA gives researchers an insight into the regulation events that are critical for many biological processes and disease states. So by combining a technique called chromatin immunoprecipitation, that's called CHIP, with massively parallel sequencing, that's called the technique is called ChIP-seq. You can survey the interactions between the proteins, RNA and DNA, enabling an interpretation of the regulation events that are central to many biological processes in disease states. So when we come to mRNA sequencing, this has become the method of choice to analyze the transcriptomes of many disease states. And it is much more accurate than microarrays, and it can capture both known and novel features. It can be applied to a wide range of species. Coming to small RNA. Small RNA sequencing is necessary because these are also known as non-coding RNAs, and they act in gene silencing and the post-transcriptional regulation of gene expression. Now coming to the types of sequencers. Illumina has a range of sequencers from benchtop sequencers to production scale sequencers. And here in the slide, as you can see, when the output of the sequencers increases from 1.2 GB with the ISEC 100 to 6,000 GB with the NovaSeq 6000, you can see that the prices of these machines also increase. Here the ISEC 4000 and the HiSeq X have been temporarily discontinued. Coming to the benchtop sequencers, the HiSeq 100 is considered to be the most affordable sequencer, and these are used for many small-scale applications. The Illumina sequencers include the HiSeq 100, MiniSeq, the MySeq sequencer, the NextSeq 550, and the NextSeq 2000. And coming to the production scale sequencers, these sequencers are used for high throughput applications and for larger studies, such as the NextSeq 550 series, the NextSeq 2000, and the NovaSeq 6000. And the HiSeq 3000 and the HiSeq 4000 systems have been discontinued. These were used for production scale, high throughput exome and transcriptome sequencing projects. Now, coming to an important parameter that is sequencing coverage. And this is something that this is a formula that needs to be known. So here, what coverage of the human genome will one get with one lane of a high-sec 100 in paired-end mode? That is 2 into 100, if 200 million clusters were bound. So this formula is used where 200 million represents the number of clusters, 2 represents being paired-end, 100 is the read length, divided by 3.2 GB, which represents the human genome size. And after computing, you'd get a 12.5 coverage. So this is an important term that is that one needs to know in NGS. So coming to the ISEC 100, I will be talking about these sequencers in more detail. Coming to the ISEC 100, the system specifications for this sequencer include a maximum output of 1.2 GB. It can perform 4 million reads per run, and it has a maximum read length of 2 into 150 base pairs, and 2 represents being paired end. And the applications for the ISEC 100 include small whole genome sequencing of bacteria and viruses, then targeted gene sequencing, targeted gene expression profiling, and microRNA and small RNA analysis. Coming to the next sequencer called the MiniSeq, this is a relatively inexpensive sequencer, and it can be used for simple data analysis for everyday users. It supports a range of targeted DNA and RNA applications. The system specifications for the MiniSeq include an output range of 1.65 to 7.5 GB. It can perform 7 to 25 reads per run, and it has a maximum read length of 2 into 150 base pairs. The key applications of the MiniSeq include targeted resequencing, small whole genome sequencing, and targeted gene expression profiling. There are more applications with the MiniSeq, such as tumor profiling, germline mutation analysis, small RNA discovery and profiling, 16S metagenomic sequencing, and genotyping by sequencing. Coming to the MiSeq sequencer, this has an output range of 540 MB to 15 GB, and it can perform 1 to 25 million reads per run. It has a maximum read length of 2 into 300 base pairs, and it supports a range of applications. It's a very versatile sequencer, such as small genome sequencing, 16S ribosomal RNA sequencing, targeted DNA sequencing, targeted RNA sequencing, small RNA sequencing, human leukocyte antigen sequencing, and pre-implantation genetic screening. Coming to the NextSeq 2000. The NextSeq 2000 has several system specifications like an output range of 40 to 300 GB. It can perform 400 million to 1 billion reads per run, and it has a maximum read length of 2 into 150 base pairs. The main applications for this are, it has a vast number of applications, and these include small whole genome sequencing of microbes and viruses, exome and large panel sequencing, targeted gene sequencing, single cell profiling, transcriptome sequencing such as total RNA sequencing, mRNA sequencing, gene expression profiling, targeted gene expression profiling, and ChIP-seq, that's the interactions between the proteins and DNA, and methylation sequencing, 16S metagenomic sequencing, cell-free sequencing, and liquid biopsy analysis. Coming to the last sequencer, that's NovaSeq 6000. The system specification for this sequencer include an output range of 4,800 to 6,000 GB. It can perform 32 to 40 reads per run, and it has a maximum read length of 2 into 250 base pairs. So the main applications for this sequencer include whole genome sequencing, whole exome sequencing, whole transcriptome sequencing, and it's mainly used for very large whole genome sequencing like humans, plants, or animals. In conclusion, I'd like to say that this next-gen sequencing methods are used for the identification of genes that cause rare and severe Mendelian diseases, and it can also help us understand the pathogenesis of cancer and find new ways of treating cancer. And the Illumina platforms are the most preferred by researchers probably because they have low they're more affordable than others. So with that, I come to the end of my talk, and thank you for listening.
Thank you, ma'am. Thank you for that brief and informative presentation.
Okay.
We have a query, ma'am, from Neha Singh. What is the significance of... Yeah, I'll read out, ma'am.
Okay, one moment. Just come back to them. Yeah, okay. What's the question?
What is the significance of coverage area? Why only the human genome was taken as reference?
Why was the human genome taken as a reference?
Yeah.
What? I didn't understand the question actually. Sorry. What is it?
What, ma'am? First query is like, what is the significance of coverage area?
The coverage area is necessary. We want to know how much of the genome has been sequenced, and we compare it with the reference genome. So coverage is just basically that parameter to compare it with the how much of that sequence was sequenced, fragment was sequenced.
Okay, ma'am. Next query is like, why only the human genome was taken as reference?
It depends on the human. It's not just the human genome. You can take it for any kind of, suppose it's bacteria or viruses. Coverage is, it is not just for the human genome, no. Hello?
Yes, ma'am.
Yes.
Okay, ma'am. Hello?
Hello?
Yes, ma'am. Can you hear me?
Yes, I can hear you, yes.
Okay, so shall we move on to the next query, ma'am?
Yes, yes, sure.
Okay. Next one is from Prashant Kumar. The query is only P5 and P7 adapters present, or any others can be used?
Only the P5, as far as I know, it's the P5 and P7 regions that they use. There are no other adapters. Yeah.
Okay, ma'am. Next query from Dr. Aruna Patri. Can you please tell us about sequencing using Affymetrix?
Sequencing using, I don't have that much knowledge about Affymetrix. I'm so sorry. I know about Illumina. I've done more on that.
Okay, thank you, ma'am. We have one more query from Dr. S. Selvaraj. He's asking, like, you said somewhere about total RNA sequencing. Can you please elaborate it?
Total RNA sequencing?
Yeah.
That's like for transcriptomes, for the to understand the transcriptomes of these. That's what you were asking? Total RNA sequencing, yes.
Yeah. So Maithili madam, so I wish to know. So if I'm correct, like you said somewhere about this total RNA sequencing. So can you just tell me, like, for
No,
example?
I was talking about this. You're talking about the applications, right?
Okay.
I was talking about the protein, RNA, and DNA interactions between them, and that can be found out by sequencing the RNA and DNA. And
Okay,
I was
Maithili.
talking also about chipsec. Yeah.
Okay. Thank you, Maithili. Maybe, like I was mistake because, like I was having small confusion whether can we go directly. Yeah, I got it. Thank you,
So,
Maithili. Just
okay.
Thank you, ma'am.
thank you.
Yeah, thank you, sir. Ma'am, next query from Monica Soni. Can you please enlighten us on meta-transcriptomics?
Transcriptomics. You mean to say metagenomics? Yeah. That is mainly metagenomics is used for the bacterial, to understand bacterial 16S RNA from different species. So I was talking about that here. I don't think I spoke about anything else. Yeah.
Okay,
I spoke about metagenomic sequencing. Yeah.
okay. Thank you, ma'am. One more query from Shamal.
Yes.
What is the percentage of accuracy of these machines?
Illumina has only about 1% inaccuracy. It's very accurate for most parts. It's one of the most accurate machines, and they also do Sanger sequencing to validate these results. So.
Okay, thank you, ma'am. Thank you so much. I think that's all about the queries. Thank you so
Okay.
much for joining us and sharing your ideas, ma'am. Thank you.
Thank you.
So on that note, we come to the successful completion of the 1-week online international faculty development program on recent advances in next generation sequencing. We would like to have a live feedback session of our participants on this occasion. I request some participants to volunteer and share your views on the program, please. You can unmute yourself.
Good
And
evening. Audible? Yeah, audible my voice? Madam?
yes, sir.
Hello.
Yes, sir. Yes, sir. We are hearing your voice, sir. You can proceed, please.
Yeah. Good evening to everyone. I am Dr. P. Venkatraman from SRM Medical College Hospital, SRM IST. I have attended all the sessions in FDP on next generation sequencing. All the sessions were informative and all the resource persons given excellent presentation and easily understandable also. Especially today's presentation, Dr. Maithili Verma from UK. Yesterday, Dr. Sushil Kumar, and day before yesterday, Dr. Bushmanathan Muthurulan from Harvard University, Cambridge. His way of presentation is very excellent. So for this, I thank Dr. Trinyana Sambandam and Dr. Kumar from the Department of Wash and Research, Adivama IST. Last week, the same team organized a FTP on mass spectrometry and thermal analysis. All of you know that program also very informative. So within short period, the team organized another one excellent FTP on next generation sequencing. So I congratulate the organizing team for organizing those successful FTPs continuously. Also, I wish the team for the future success. Thank you, one and all.
Thank you, sir. Thanks a lot, sir.
Thank you. Thank you.
Thank you so much, sir. We would like to have a few more feedbacks from the participants.
Hello.
Yes, ma'am. Please talk, ma'am.
Myself, Dr. Jasmine from Zoological Survey of India. This is the first time I am attending one of the webinars of the Satyabama Institute of Science and Technology. And I have seen in the Facebook the details, and then I think Dr. Imbakandan is one of my friend. I think through that I have got a shared post, and I have attended. It was this webinar. It is a very excellent webinar, and also that most of that because I am basically an ecologist. This is the first time I am attending a molecular level webinar or a presentation. So it's like an excellent all the resource persons, they have given excellent presentations, and in a very basic level to, I think some of today Maithili's Illumina sequencing, it is in my opinion it should be in the second or third, because in the first three presentations were the more the level of my understanding is little bit high in that, but here this one is some more basic, so it should be in the first, and the remaining thing should be in the following. Then it is very easily understandable for as a means in the molecular level basic researcher. So all the presentations are excellent, and the previous feedback he has given that Dr. Pushpanathan, he was like a teacher. He has explained everything very nicely, and first and Dr. Maithili also has given in that way. So I am very happy for attending this webinar and these things, and I think I also thankful to the organizers, Dr. Imbakandan, Tilubranazam, and all. And thank you very much. Thank you very much. And I am expecting more good webinars from the Ocean Research Center of Satyabama. Thank you very much. Because what I am expecting, I have got more than that.
Thank you.
So I will suggest to my students also and other colleagues also. Thank you very much.
Thank you.
Thank you, ma'am. Thanks a lot, ma'am.
Thank you so much.
Hello, good evening, everyone.
Good evening, sir.
This is Prashant Kumar from Saptagiri College of Engineering, Bangalore. In fact, very in a short notice, I received invitation from one of my friend. So I just entered to this Satyabhama's international FDP program. See, we have got lot of things. In this COVID crisis, many FDPs I have attended, but the major breakthrough with this Satyabhama was they were very punctual. 6 o'clock on dot, 6 o'clock they used to start the program. You used to start the program. So that really made. And similarly, no much extension. 7:30, within 7:30 on dot, the program used to get over. So it really scheduled us. And the speakers, especially today, Madam Maithili was excellent. Similarly, yesterday, Dr. Sushil Kumar Midda, because I know him personally, and he was also excellent. Similarly, all day 1 to now, 7 presentations were chosen most importantly, and it covered almost all the areas of NGS process. So similarly, I would be very happy to join for Satyabhama's further FDPs or webinars, because I really they have made it possible. Each and every, as Madam was saying previously, entire process, entire speakers who delivered the lectures, they were excellent, and at a ground level, they were able to induct the knowledge into our brains. So thanks a lot, Center for Ocean Research and Thiru sir, for making it possible and giving us a platform to witness the most eminent speakers. Thank you, sir. Thanks a lot for the organizing committee and
Thank
all
you.
the
Thank you,
speakers
sir.
who made it possible.
Thank you, sir.
Thank you.
Thank
Thanks
you,
a lot,
sir.
sir. Thank you.
Thank you.
Hello. Hello.
Yes, sir. Mohammed sir, you can please, sir. Yes, sir.
Okay. I'm Dr. Mohammed from Veterinary University, Kerala. Actually, I first heard about Satyabhama University from Imbakandan sir. We attended a ICR training together at CMFRI Cochin. Now I feel that that meeting, since we met together some 1 year back, was a very fruitful one, because all the sessions we attended, I also attended all the 7 sessions, as other participants, my colleagues told. The faculties they were actually not taking a class, but they were sharing their experiences, or otherwise they were just instructing what they were doing in their lab. Because while attending the class, I felt that I was just working in a lab. That was the feeling I felt. So my humble request to Imbakanthan sir and other team is that you please enable us to continue this link or the collaboration, or whatever, maybe the network, because all those faculties and the participants, even the interactions were very much effective. Means in many cases, when I started asking questions or when I was typing questions, some others had already asked that question. Means that is actually the impact of the class, and the wavelength of the participants also were one and the same. So my request is that please enable us to continue this network by continuing this WhatsApp group or other networking. And thank you very much, Imbakanthan sir, and all other organizers for arranging this webinar in a very effective manner. This also gives us the confidence to arrange such sort of seminars or webinars in our institution also. Thank you very much. Thank you very much, all my colleagues and organizers and faculties. Thank you very much.
Thank you, sir. Thank you so much.
Thanks a lot, sir. Thank you. Any other participant willing to share their review here? Namita?
No, sir. I think that's all.
That's all.
Okay.
Okay, so yeah. Thank you. Thank you all for your feedbacks, and I now request Dr. Imbakanthan, Head and Scientist, T Center for Ocean Research, Satyabama Institute of Science and Technology, to deliver the vote of thanks.
Thank you, Namita. Thank you, Thiru, and thank you, Kumar, for this making this session fruitful. From the review of all the participants, I am really happy that our team is working well. And a warm good evening. First of all, I forgot to even say good evening to everybody. With the blessing of our beloved founder chancellor, Dr. JPR sir, actually I am very happy to share the vote of thanks here on behalf of Center for Ocean Research. We sincerely express our heartfelt thankfulness to our Honorable Chancellor, Ma'am Dr. Maria Zina Johnson, and our President, Sir Dr. Maria Johnson, for their constant guidance and support and encouragement in all our activities. It gives immense pleasure to thank our Vice Chancellor, Ma'am Dr. T. Shashiprabha, and our Director Research, Ma'am Dr. B. Sheila Rani, for their valuable presence in the session. Even in the inaugural session, they are there with us, and even some of the sessions they have attended, they have monitored us, and they have informed me through phone. Thanks for their support, and I wish there's support from both of them in continuing all the sessions in Center for Oceans, whatever we are organizing. Coming to the speaker, and I really thank the wonderful sessions, whatever delivered by the speakers. It's the right time I have to thank Mr. Moonak Shabda, National Center for Biological Science, Tata Institute of Fundamental Research, Bangalore. Mr. T. Sendal Kumar, Applied Genomics, Bangalore, who is my good friend. Dr. V. Naveen Kumar, Immunogenics Private Limited, Chennai. He is also one of my good friends. Dr. Pushpanathan Muthuralan, Harvard University, Cambridge. and Dr. Sushil Kumar Nidula from Mahalakshmi College for Women from Bangalore, and today's speaker, Dr. Maithili Verma, University of Essex. And I think I have missed Dr. Ogli. I know Ogli recently. I'm a friend. I'm friend of him in Facebook, and even Dr. Dinesh from NIO, he narrated more about him. Actually, Thiru is conducting this program because he is one of the person who is working in genomics in our Center for Ocean Research. And we at Center for Ocean Research, we are working and concentrating on elasmobranch mitochondrial genomics, marine mitochondrial genomics, fish gut microbiome metagenomics, and seaweed transcriptomics. Apart from that, we are contributing more than 650 short 16S rRNA sequence in NCBA related to marine microbial sequences. And as Dr. Mohammed said, we want to continue this network. Actually, I request Mr. Thiru as well as Kumar to make a WhatsApp group, and you have to unite all of us, including please include me also in that group, as well as the speakers and the participants, so that the network will help us. And I wish all the participants as well as the speakers, if you have a chance to come to Chennai, please visit our center and please interact with our conveners, co-convener, as well as to our scientists who are all working in Center for Ocean Research. Thank you. Thanks a lot.
Thank you, sir. Thanks a lot, sir.
Thank you, sir. Thank you, sir.
Thank you. Thank you. Thank you very much.
Regarding the feedback form and all, already you have shared.
So, shall we end, sir?
Yeah, shall we end the session? You have to, have you shared the feedback?
Yeah, already we shared the feedback link, sir.
Okay.
And certificate will be issued within 3 days.
Yes, sir.
Okay, okay, okay.
Certificate and all the PPT and recording videos will be sent within 3 days.
Yes, sir.
Okay, okay. Thanks to all the participants, and thanks to the today's speaker, and thanks to everybody.
Thank you.
Thanks a lot.
Thank you, Maithili. Thank you, Maithili.
And thank you, Namita, for being moderator of the session. Thank you.
Thank you so much, sir. Thank you.
Thank you.
Thank you all.
Thank you. Thank you all.
Thank you, Maithili, ma'am. Thank you.
Thank you.